PCR and electrophoresis. Common lab techniques to amplify and recognize specific DNA sequences. PCR reaction. The polymerase chain reaction is a molecular biology technique used to amplify in vitro a single copy or few copies of DNA. Taking into account, we have to know partially the sequence of the fragment we want to amplify. For example, a gene, a part of a gene, a non-coding region. Type of PCR: quantitative PCR, reverse transcription PCR. First, we will talk about normal PCR. Component needed: DNA template, the sample DNA that contains the target sequence, DNA polymerase enzyme that synthesizes new strand of DNA complementary to the target sequence, primers, short piece of single-stranded DNA which are complementary to the target sequence, nucleotides. PCR reaction occurs in this type of machine. The steps: initiation steps, denaturation step, annealing step, where primers anneal to the single-stranded DNA and incorrect annealing temperature will cause an error. Elongation step, polymerase synthesizes a new DNA strand complementary to the template strand by adding DNTPs in 5 to 3 direction. Final elongation, final hold for indefinite time. The processes of denaturation, annealing and elongation constitute of one cycle. Multiple cycles are required to amplify DNA. Quantitative PCR. We use this PCR variant to quantify DNA. It's amplify and quantify at the same time. Tagman Prof. Components: conventional PCR materials, DNA fragment, DNA polymerase, primers. Prof. In the prof, one of the nucleotides is marked with a fluorescent molecule, which is called fluorophore, and with other that absorb the light emitted by the fluorophore, known as quencher. This will happen only if they are together. When they become separated, fluorescence will be off. Detector. Procedure: initiation, denaturation, separation of the double strand, annealing of primer and Tagman Prof, elongation. DNA polymerase add nucleotides from 5 to 3N. The 5 to 3 exonuclease activity of the Tagman polymerase degrades the prof that has annealed to the template. The close proximity between the quencher and the fluorophore is broken. Fluorescence is produced. Silver green proofs. Other kind of quantitative PCR is based on silver green proof. Silver green is a molecule that emits fluorescence when bound to double-stranded DNA. This dye is used to track the progress of DNA amplification in real-time PCR. In the exponential phase of PCR, the amount of DNA increases exponentially, and so does the fluorescence. The fluorescence emitted is proportional to the amount of DNA. We can use a known amount of the standard DNA sample and measure at which cycle the fluorescence increases above the background. Analysis of results. The result of a quantitative PCR is expressed in an amplification graph. The fluorescence emitted, which is directly related to the amount of DNA amplified, is plotted against the number of cycles of PCR. The amplification curve obtained consists of: initial phase, the fluorescence production is below the detection level of the thermal cycle. Second phase, the fluorescence production increases exponentially. Third phase, the reaction is finished and the fluorescence production reaches stability. The main values that can be extracted from the graph are: threshold line, it defines the value at which the exponential increase at fluorescence production starts. Quantitative cycle, point at which fluorescence becomes measurable. The higher amount of DNA initially, the sooner this value will be reached, since a lower number of cycles would be needed. But how much DNA do I have? To quantify the amount of DNA that has been amplified, a calibration curve will be created by means of different known concentrations of DNA sample. The CT value could be inversely proportional to the initial DNA concentration. Afterwards, the CT value obtained are plotted against the initial number of copies per microliter. The straight line obtained will allow to calculate the amount of DNA amplified in the sample study, just considering the CT value calculated. Reverse transcription PCR. First, uses. It is used to detect genes expressions through creation of cDNA from RNA. Second, materials. RNA template retrotranscriptase PCR materials. Third, procedure. The RNA template is first converted into complementary DNA using a reverse transcriptase. The cDNA is then used as a template for exponential amplification using PCR. And after the PCR, it comes an electrophoresis. Why an electrophoresis? In order to verify the PCR reaction and to find out the approximated concentration of our sample. Materials. DNA sample, DNA standard, electrophoresis equipment, sodium hydroxide, UV light, ethidium bromide. Procedure. First, denaturalize DNA sample using sodium hydroxide. In that way, the form of the molecule won't affect the movement through the gel. Then dye the sample using ethidium bromide to allow us visualizing it under UV light. Then load DNA sample and standard DNA into precast wells in the gel. Why standard DNA? Well, because standard DNA contains a mixture of DNA fragments of predetermined size that can be compared against our sample to estimate the size and concentration. 4. Run the gel. In other words, run the electrical field. 5th, see our sample on the UV transilluminator.